Good morning to all of you. Today we are going to discuss transmission of impulses. So how transmission of our impulses work in our body. So first assume that. How transmission of impulses occur in our body. If you touch a hot object, you feel that this object is hot. How? So our body made up of the signal that can be transmitted through the nerve. So this nerve from the one part to another part, the brain, which can sense the all kind of changes, and these changes or this information are transferred by the nerve. So how the nerve help to the transmission from the one neuron to the next neuron and simultaneously go on. So this process that is called transmission of impulses. So next you have to understand that synapse. Synapse mean the junction between the two neurons. So here the junction, it means the presynaptic neuron, and this neuron transmit the signal to the next neuron, that is known as a postsynaptic neuron or nerve. So ultimately conclude that this synapse, it means the junction between the two neuron, and by the help of this junction the impulses transmitted from the one neuron to the next neuron. And this impulses or synapse that can be formed by with the help of the axon and axon, presynaptic and postsynaptic. It can be form a synapse if the both part are axon. It can be form a synapse. Second one can form one part presynaptic neuron, that is axon, another part that is dendron, that can also form the synapse, and third type of synapse can be formed by the axon and cell body. So here is the axon part and cell body. These kind of synapse known as a axosomatic synapse. So based on their parts, it divided into three type of synapse: axonic synapse, axodendrotic synapse, and axosomatic synapse. Okay. Next, we are going to understand the type of synapse. This type of synapse means the synapse, it based on their, it release the chemical or their bind firmly. On the basis of that, it divided into two types. One is electrical synapse, another is a chemical synapse. Electrical synapse, it means there is a no gap between the presynaptic neuron and postsynaptic neuron. It means the both part of the neuron, they are joined firmly. There is a no gap. It means the signal, it can transmit very quickly. Okay. And second type synapse means those synapse, it can transmit the signal through the chemical. Through the chemical means it release the neurotransmitter from the presynaptic neuron, and it can inhibit or excited the postsynaptic neuron. So these kind of synapse, which is known as a chemical synapse, it means release the chemical from the presynaptic neuron, and it can excited postsynaptic neuron, it can be inhibited postsynaptic neuron. So based on that, it divided into two types: synapse, electrical synapse and chemical synapse. Okay, next come to the structure of synapse. What is the structure of synapse? Synapse, we have already understood that it means the junction between the two neurons. One must be the presynaptic neuron and another is a postsynaptic neuron. So here is the structure: the end part, the terminal part of the axon, and the cell body of the neuron. So here is that is the presynaptic membrane, and on the opposite side, which is known as a postsynaptic membrane. And the junction between the two presynaptic membrane and postsynaptic membrane, this gap is called synaptic cleft. So here the presynaptic neuron, it means when the signal comes into the end part, this membrane, which is known as a presynaptic neuron or presynaptic presynaptic nerve, and this part also known as a postsynaptic nerve, and the membrane which is known as a presynaptic membrane and postsynaptic membrane. Okay. That how the process of transmission of impulses occur in the synapse part. So first, when the signal reach to the presynaptic nerve part, so due to the changes of the sodium and influx of the potassium ion in the membrane of nerve, when it reach at the end presynaptic nerve, that time the neurotransmitter accumulation in the part of the presynaptic neuron or membrane, it's near about presynaptic membrane, the all neurotransmitter are accumulated, and next process, the calcium ion which is present in the synaptic cleft. So calcium ion enter through the presynaptic membrane, which help to the increase the permeability of the presynaptic membrane. So what is the role of the calcium? This calcium which present in the synaptic cleft and it enter inside the presynaptic nerve part, which increase the permeability of the presynaptic membrane. And if the increase of the presynaptic permeability of the presynaptic membrane, this neurotransmitter become closer to the presynaptic neuron, and ultimately it fuse with the membrane, and the all neurotransmitter get closer and it release their neurotransmitter. And this neurotransmitter are different type. So it can be excitatory, it can be inhibitory. Suppose in case of excitatory, that is ACH, acetylcholine. This acetylcholine only plays over the acetylcholine receptor. It cannot place over the another kind of receptor, so it is very specific. So all kind of receptor present in the postsynaptic membrane. So if neurotransmitter release the acetylcholine, this acetylcholine plays over the acetylcholine receptor in presynaptic membrane. So when it plays, it excited the membrane. If the another type, that is known as a GABA, GABA, which is an inhibitory neurotransmitter. If inhibitory neurotransmitter plays over the GABA receptor, it can inhibit the process of the signal. So it can be stimulate the next neuron, or it can be inhibit the next neuron. So depend on the neurotransmitter, it can be excitatory or it can be inhibitory. So inhibitory receptor, it prevent the signal to the next neuron, and excitatory, it help to the transmit the signal from the one neuron to the another neuron. That is the mechanism. So when the calcium enter into the presynaptic neuron, it increase the permeability, and then neurotransmitter vesicle which come closer to the presynaptic membrane, and it fuse with the membrane and release the neurotransmitter. And these neurotransmitter are specifically bind with their respective receptor, and that help to the either excitatory effect, either inhibitory effect. After few milliseconds, this effect already done. So this neurotransmitter must be removed from the receptor, otherwise it will give the continuously signal for the excitatory or inhibitory. So their respective esterase enzymes. Suppose in this receptor, acetylcholine plays over the acetylcholine receptor. So this cholinesterase, which is a kind of enzyme which break down the acetylcholine to free the receptor of acetylcholine receptor. So it ready to accept the another neurotransmitter, acetylcholine neurotransmitter. So this acetylcholine esterase which break down the acetylcholine neurotransmitter for the maintaining the signal process. And the last, ultimately it help to the again it exchange the sodium and potassium ion in the part of the postsynaptic neuron, and that help to the transmission the signal from the one neuron to the another neuron. Let's try to understand the neurotransmitter, which is a two type. One is the excitatory neurotransmitter and inhibitory neurotransmitter. So how this excitatory neurotransmitter work in the synapse part, and how the inhibitory neurotransmitter act on the synapse part. First, if excitatory neurotransmitter, one of the example we have already understood, that acetylcholine. When acetylcholine fused with the presynaptic membrane, released in the synaptic cleft and placed over the acetylcholine receptor, it help to the excited the postsynaptic neuron. So it means the sodium enter through the postsynaptic membrane. So already we have know that normal resting membrane potential of the nerve, that is minus 70 millivolt. So due to the influx of the sodium inside the membrane, it reach minus 55 millivolt. So when it reach 55 millivolt, it reach threshold stimulus. So again the process of the exchange sodium and potassium, it continuously start through the membrane. So by this process the signal which transfer to the next neuron. So that is the mechanism of neurotransmitter, that kind of excitatory. If the inhibitory, how it work? Suppose the here is the example is a GABA. Gamma-aminobutyric acid is a one kind of neurotransmitter. When this neurotransmitter fused with the presynaptic membrane and releasing synaptic cleft and place over the GABA receptor. So this GABA receptor what it do? Influx of the potassium. So due to the influx of the potassium, so the normal resting membrane potential, that is minus 70 millivolt, but due to the influx of the potassium, it again reach minus 85 millivolt. It means that is a condition hyperpolarized state. So in this state, the action potential cannot be generated. So ultimately, so the process of generation of the action potential that are not done in case of inhibitory neurotransmitter. In case of excitatory neurotransmitter, this process are continuously transferred to the next neuron. So if I summarize the whole thing, so we have understood the synapse in the junction between the two neuron. Next we have understood the type of neuron. It means that based on their release the chemical or their bind family, it depends on that. There is a two type, chemical synapse and electrical synapse. Next we have understood the structure. Here is a presynaptic knob, postsynaptic knob. This presynaptic knob consists of the presynaptic membrane and postsynaptic knob covered by the postsynaptic membrane. And in the presynaptic knob, in this area, vesicle form, that is neurotransmitter present inside the vesicle. When the signal comes, the vesicle of the neurotransmitter that are accumulated in the presynaptic knob part, and due to the influx of the calcium ion, it increases the permeability of presynaptic membrane, release the neurotransmitter and place over the receptor which present in postsynaptic membrane, and based on their chemical neurotransmitter, it can excite the process or it can inhibit the process. So that is the summarize of all the whole things. So I hope you like that. Thank you.